take the time to go through how those methods are done, the strengths and weaknesses of those methods, the perception of the strengths and weaknesses of those methods. And then we want to take a look at a really interesting testing study that just came out where a bunch of healthy volunteers exposed themselves to COVID.
Well, we're exposed to COVID.
Well, they agreed to it. They did it to themselves. Actually, 25,000 people volunteered, and they chose 36 people, 2 of which got disqualified at the last minute. And they had some really striking findings about it. And so after we look at that, I think it will allow us to reevaluate the detection methods that we went across in the first part of the class. So we're going to try to split this lecture into those 2 parts. You guys are lucky because Dr. Bennerberry and I have done thousands upon thousands. Well, that's probably exaggerating. We've done a lot of PCR tests in Qpcr. And I suspect if any of you go into medical research in any degree or biology, you will be doing lots of PCR because it is essentially as they say in many of the papers over and over, it's the gold standard of genetic testing, right? So,
To that, we are going to read a paper that appears to absolutely love PCR.
It does, and I'm not super persuaded that this is the best testing method after reading this paper, but let's go through it and see if y'all are persuaded, and then we can talk about it afterwards.
So, before we do that, I have a rundown of all these different methods in the presentation.
Yeah. From this paper?
Between the 2 papers, I pulled a bunch of...
So there's basically 3, right?
There's also Elisa's, and they talk about like various other stuff.
This, the paper, I don't want to get too bogged down in the details of the back half of it because there's essentially a lot of different ways that you can speed up PCR and speed up rapid antigen tests.
And make it cheaper and make it more accessible.
Yeah,
and
super exciting.
A lot of it is kind of technologically important, but not necessarily as interesting as the second paper.
Yeah.
So to start with, when we're talking about COVID-19 spread and testing, we have to be able to understand the assays that they're using in these papers and the point of each assay. So in the paper about infecting healthy volunteers, they keep talking about this focus-forming assay. They're comparing...
Can we get to that paper?
We can save it, yeah. Okay, that's a good point. So they're using focus-forming assays, which we'll go over when we get to the second paper. They're using ELISAs. They're using all kinds of different techniques, but they're also using this technique called Qpcr. And Qpcr is what the methods paper is really focused on because it's been used as the standard form of COVID-19 testing all around the world. And anytime that you talk about this, you will hear people talking about things like cycle number, CT thresholds, and what does that mean? How is this related to a positive test or a negative test? And what is a Qpcr versus a regular PCR? So Qpcr is basically just a quantitative PCR test, which is that based off of the way that you set up the reaction, you are going to be able to tell exactly how much genetic material you have in your sample, which you then should be able to translate back to how much of your initial inoculum you had when you started the test. And that means that you should be able to translate that to roughly how many viral copies somebody is carrying, which then should be able to translate to how sick or how infectious this person is. Whether or not that assumption holds across all cases, we're going to get to in the second paper. But here, basically, there's 2 different ways that Qpcr is diagrammed out, which is the fact that you either have something that is a 1-step or you have something that is a 2-step. And a 1-step process does the reverse transcriptase part of Rtqpcr, and it also does the actual quantitative analysis. The one that's 2-step, you do first you create complementary DNA, and then you use that because we can't amplify RNA the same way that we can amplify DNA. So all Qpcr, if it starts with RNA as a material, is always Rtpcr as well, because you have to reverse transcribe your sample in order to get this like happy, melting and annealing and copying to work. The same way that with generic PCR, you would use primers. Those primers attach at specific sites in the DNA, and then you get amplification. The same thing happens with Qpcr, but with Qpcr, you have to have something else, which is that you have to have a standard, because the standard is telling you how much DNA there actually is, and so you compare your experimental sample against this tightly titrated group of samples. And so here on the right, we see what this actually looks like in the device. So down on the bottom, you have cycle number, and on the left, you have relative fluorescence. And there is something that is the baseline fluorescence as is measured in your sample. So you mix all of your stuff together, you put it in the Qpcr machine, and it's shining a light through each well. And it's just like, okay, nothing in this well, this is our baseline. And then as you go through the process of...
Hold on, let's talk about that really quickly, just the basic detection thing. So the way that fluorescence works, if you're not familiar with it, is you shine one color of light on something, and if it's a fluorescent molecule, the molecule changes shape, and it gives off a light of a different wavelength. And so that's what Dr. Penneberry means by you shine light on it and nothing happens.
Yes. You just can't detect anything. I'm looking up to see if I can get a good image of this right now.
Just do IHC or something.
Oh, is this it? Basically, so but this is the opposite. Perhaps this is not the time to be doing this. We shall return to this. But basically, the way that fluorescence works is that you include a chemical in this reaction that intercalates into DNA, which what that means is that it is found in between all double-stranded DNA. And so as your amount of double-stranded DNA increases, you get an increase in your total fluorescence, because only double-stranded DNA will fluoresce. And you have your control, which is a sample that you know exactly, it's in a different set of wells. You know your primers, you know your template concentration that you start with, and you make a dilution of your template concentration, and this is what it's showing here in these different colors, right? So you have this no template control, which is in gray, and it doesn't fluoresce at any point. And then as you increase the amount of, as you decrease the amount of template that is inside your sample through a series of serial dilutions, you see that the point at which you start to rise above your baseline fluorescence of nothing gets later and later and later. And this makes sense, right? Because since PCR is a doubling process, if you start with one template, you're going to get above your thresholds slower than if you start with 10 templates, or if you start with 100 templates or 1,000 templates.
And I think it's really worth like putting this into real concrete terms. So like the difference between 24 cycles and 40 cycles.
40's over here.
Yeah, 40, this is like this
10
one,
to
right?
the 1.
10 to the 1 versus 10 to the 5th, 6th. I mean, let's say the purple one. The difference between 20 cycles and 40 cycles is 1 million copies of the thing. So these are like huge amounts. So the longer that you cycle something, you can detect the very, very few copies, essentially. This seems like 10 copies.
Yes, so basically at 40 cycle, you detect roughly 10 copies.
And this is after amplification.
Yes, after amplification. And this is your, because if you have less than 10 copies that you start with, then amplification doesn't get you above your baseline of fluorescence, even at 40 cycles. Maybe it will if you go far beyond that, but generally you're not using PCR to isolate. You're not looking for a single sequence. You're usually using PCR, and you're putting in a significant amount of your template. Like you're putting in, micrograms of DNA to start with. When you look at Qpcr, real Qpcr curves, and I pulled this from a paper which is down here, which is on a different virus. But basically, you see that you have these really nicely grouped standard curves. The reason the standard curves are grouped like this is because when you design a Qpcr, you replicate each well. So biology is really messy, and so what you do is you have both biological replicates and technical replicates. And in order to get a really nice control curve, you have a lot of replicates. You have usually about 4 times that you repeat the exact same reaction before you move to a different dilution, because that way you're basically demonstrating that your experimental technique is like neat and nice and tight. But what you can basically see is that in a real life PCR, and this is like a relatively clean Qpcr, you have to have this nicely fitted standard curve where you can graph your concentration relative to the CT cycle. And the CT is the cycle threshold, which is basically a point upon which you begin to see an inflection in this curve where the curve starts to rise and it's just about to go linear. And that's the place where you set your threshold. And based off of that threshold, you look to see, okay, how long does it take? How many cycles does it take for me to hit this threshold based off of my dilution? And somewhere along this, you will look at each one of these individual messy samples, see where it rises, meets the CT threshold, and then you'll tell yourself, okay, this is the amount of genetic material that I have in this reaction, and so I can extrapolate backwards to how much I had when I started, and that's the quantitative aspect of Qpcr. A normal PCR, you don't have any of these standards. You don't run standards except for yes, no, is there a reaction that happens? And so what you can see is that there is a difference between a strong positive and a weak positive. And that has to do with cycle number, where when you have a strong positive, it rises above background fluorescence earlier than the one that rises not as strongly above background fluorescence and does so much later. ELISAs we'll talk about when we get to the antigen portion. Lateral flow is also antigen. We'll get to all that. There's a couple of other really interesting diagnostic tests that before we get into the paper I wanted to mention. One of these was Sherlock. So Qpcr is kind of the world's standard. Sherlock? Yeah, that's why I pulled it. So Sherlock is a really novel way of doing sequence detection. And it basically has to do with being able to, amplify your sequence the same way that you would with PCR, but then your detection method isn't an Rtpcr machine. It isn't any kind of mass spec. You're not actually physically quantifying the amount of DNA that you have there through an external method. You have an enzyme that is inside of your solution that is able to cleave a special reporter and release light. So it's a light-based method still, but it's using this Cas13 protein, which was recently discovered to be very effective at cleaving DNA, and so people are now trying to figure out ways that you can kind of like include it in all kinds of other things. I just thought that was kind of cool. And then we'll get to that later. Okay, paper time. So we need to switch the share. Okay. Current state of diagnostic, screening, and surveillance testing methods for COVID-19 from an analytical chemistry point of view. Where should we start?
We can start right at the beginning. I think there's like some interesting points here.
All right.
The thing that I think is interesting is like comparing both these two papers together mostly. I was kind of reading them at the same time, and there's like a bunch of really interesting contradictory information between the two. right out of the gate, they're telling us stuff that we've heard a million times, but I think it's kind of the wording's quite interesting here. A seafood market in the Chinese city of Wuhan was suspected to be the center of an outbreak of pneumonia of unknown origin. The pneumonia was later confirmed to be secondary to infection by a novel coronavirus. And then the World Health Organization named the disease COVID-19, and some committee that I didn't get a chance to look up. Did you look them up? The ICTV gave it the name that we all know and love. Symptoms can be mild or severe, but because it's very contagious, it's a threat to humanity in just a few months. And then further on in the introduction, next paragraph, the most worrying aspect is the detection of the virus transmission from asymptomatic people. What else?
They're making the case that, these analytical techniques are the best standard for being able to avoid catastrophe. And, the aim of this manuscript is to introduce, from an analytical point of view, the main coronavirus detection techniques. The gold standard method to detect SARS-CoV-2 is named Rtpcr. This is the same as Qpcr, it's just reverse transcription quantitative PCR. They're going to introduce all of these different alternatives and other types of tests to see if they're better able to confirm the infection of the virus.
I think the word infection is what's really critical here. Like, what is an infection with the virus exactly? Because I think what they're making the case for is that PCR is capable of detecting the teeniest bit of viral genetic material, right? So it says that the cycle range goes out to 40 cycles, which, according to your graph from the Qpcr manufacturers, is like essentially the tiniest bit imaginable, right? And presumably, honestly, like if you've done these before, you can cycle them out to infinity. You can detect stuff even when there's nothing there at all, right?
Sometimes it's not a guarantee. I've definitely had to go both ways. Let me tell you, I've run a lot of Qpcrs and just had them come up with 0, even in the control wells.
So I think that they're arguing that the PCR is the best test because it's the most sensitive.
But the question is, does sensitivity translate to a really meaningful estimate? And
like if your goal is to prevent sickness and death,
then
you need to be able to detect sickness
as a corollary of the amount of virus. And that's the question. Like, does the amount of virus that you have quantified in your Rtpcr test directly relate to the level of sickness?
Or infectivity or anything like that, right?
In this very small group of people who we're going to look at later, it seems to say that it doesn't. And that's a really interesting thing, because when this paper was written, this testing and detection paper, they obviously hadn't done this human challenge trial yet. And if they hadn't done the human challenge trial, then it was impossible to know that there wasn't this direct relationship between amount of COVID-19 virions present and severe disease. But now that we have this direct information, it's starting to create this really, really confusing picture, because you're looking at it and you're like, well, hold on a second. If our gold standard is a test that evaluates number of copies, and we seem to be finding that the number of copies doesn't correlate to severity of disease, then how is this our gold standard test?
Meanwhile, the antigen test in the second study detected 100% of the people who were sick by enormous margins. It was like 1,800 versus 0.
In the second,
3,000 versus 0 antigen units or whatever it was in the second paper.
Yeah, let's look at that when we get there.
I guess I'm just trying to say that the antigen test seems to be super effective, actually.
Yeah, like it makes me wonder if these people aren't somehow involved in some, like, I wonder if they have some stake in a PCR company, or they're producing some kind of PCR related product, because it's a very perplexing statement given the amount of false positives that we get off of these PCR tests. But you're welcome to write whatever you want in a science paper as long as you make your case, right? And it's possible to make a case. And the case that they make is the fact that it is the most stringent. And
which is, I'll give them that. That's definitely clear. You can definitely detect the most COVID you can definitely detect the most virus in people with PCR. But the question of whether that's a good idea, whether that's what you should be doing, is going and finding every person who has even a flake of COVID in them. I'm not sure how useful that is. Like, I'm not sure how that's useful to getting our society back on track when it's a virus that's becoming endemic to the point that we're at 85+% seropositivity. Of course, everybody's going to be exposed to it in some level or another. The goal we should be after is stopping people from getting each other sick. And so we need to think about ways to tell if people are infectious in the sense of causing disease, not infectious in the sense of transmitting test positivity to one another. That's not particularly interesting. So I don't know. I've just, it could be that it's just a late punch. This Nature paper just came out, so maybe that'll change the game a little bit. And this paper that we're looking at right now came out in 2021, I believe. So, I don't fault the authors or anything. I just think that it's a really fascinating about face. I don't know. Is there anything else to say about that? No. So let's look at the way they did the test. Dr. Penderbury kind of talked about it a little bit, but you all have probably been through this before, right? They swab the back of your nose, or in some cases they get people to spit into a test tube. And this just starts to highlight also some of the variability in this, right? We know that you get different numbers of copies of the virions depending on where you look in the oral, pharyngeal, nasal area. And so, they complain later on that there's a lot of diversity between sites for these different PCR thresholds and stuff, and it might have a lot to do with all these variable methods and primers and everything else that's going on here. So what they're going to do is they take these samples and they have primers against one of these targets. And it doesn't seem like they're a standardized set of targets either, which makes it more difficult because these targets persist at different intervals, right? I believe, without looking it up, I can't tell you which one it is.
Sorry, I kind of lost the thread. What are you saying?
The different targets, the gene products that they use as primers, they appear at different intervals in the infection course. So some of them appear early, some of them appear later.
And so is the reason that they use, so you're saying that they use different primer sets or gene, what do you mean by gene products?
The primers are directed against the gene products.
Well, they're directed
there's the genes for these protein products that turn out to be different parts here. So if we go up a little bit, so these pieces here.
And so what's the finding that you're saying?
I'm just saying this is like another chance for some heterogeneity to be introduced into the sampling process because not everybody's PCRing for the same products.
But
they appear at different time courses.
But if the proteins appear at different time courses or the genes do? Because how could the genes appear at different time courses?
That's what it says earlier though.
Where it does?
I'll find it.
Find it. All right. So while we're dealing with that question.
Yeah, the genes too. I'll find it for you.
Yeah. So basically, once you have the sample, you prep it, you add this thing that's called master mix, and then this is the reaction that I was talking about earlier. You start with this viral RNA, you add a primer to it, which is in green, then you extend the RNA using your reverse transcriptase. Your reverse transcriptase gives you cDNA, and now what you have is you have this kind of complementary DNA to the RNA, and that gets inserted into this replication cycle. And what you do is you have these probes that stick to the surface of the DNA. And when the probe is displaced off of the surface of DNA in this version, because there's other versions of this kind of thing, it gets cut by the oncoming reverse transcriptase, or the polymerase in this case, sorry. And when it gets cut, you start to see fluorescence. Fluorescence results in this increase over time, and you have your threshold where you hit the linear phase. What are you finding?
It's just that the sensitivity, depending on which gene you choose, appears at different points in the time course. So this is
So where are you at?
Yeah.
All right. The study of Reyna and Suarez proved that the sensitivity of the Rtpcr test based on the amplification of the gene E decreases in samples taken during the last period of the infection, what represents mostly patients with resolving infection. However, the sensitivity in the detection of the N gene increases in these samples. So basically, it's like it's not consistent over the time course of the infection, and if you are PCRing, if you are just targeting one of these genes, you might not be getting all of them you won't be getting a consistent reaction from time to time. And that's
and sites aren't doing it consistently too, right? So they make the case later on that we have the availability of doing multiplex Qpcrs where we can amplify multiple targets at a time. And they're like, that might be a good idea, but that's a recommendation being made in the middle of last year. And I don't know if anybody's doing it.
Yeah. And they do say here also that in general, 30 to 45 cycles of PCR are needed to detect the virus. Amplification is the limiting factor to transform the standard method into a rapid test, but it is thanks to this process that Rtpcr has such good sensitivity. Again, I think that there is a perplexing lack of attention paid to the need to demonstrate a clear relationship between number of PCR cycles to
outcome.
Outcome. Because it's not, virology is so weird and so unexplored that it's very, very easy to make assumptions based off of what we think that we know, and then to move forward and to build on them without taking the time to really analyze whether or not our assumptions are correct. And that's a hard thing to do in the sciences. It's a hard thing to do when you have methods that you think work really well, and you have methods that you base a lot of your work upon. And so
it's an interesting case of a method that works incredibly well at detecting virus, but fails to actually consider whether that's what the goal of testing should be. I hope that comes across clearly.
I mean, they show basically that, okay, so cycle threshold values of less than 37 are reported as positive results. CT values that are larger than 40 are considered clinically negative, and anything that is within 37 to 40 is dubious and the test should be repeated. So
well, that's what the Chinese CDC came up with. However, the American CDC is going anywhere between 30 and 45 cycles. Right? It's just like they're really, really proud of themselves for the sensitivity of this test. And I do think that's cool that they can detect that the tiniest bit of virus is present. But they're just completely unconcerned with whether that means you're A, infectious, or B, sick.
There's also like a pretty significant variation, right? So they say that some studies have shown that virus culture is unfeasible in cases with a CT of greater than 33, which means that if you're positive with, there's a realm of people who are positive between, let's say that if your laboratory is using cycle thresholds between 30 and 45 cycles, so let's say you're using 45 cycles. So if you're using 45 cycles and you're calling people positive, that means that a CT threshold that is greater than 33 doesn't actually culture any viruses. Like, you cannot take that sample and put it on cells and have the cells be infected, and yet you have a window where you would be reporting that as being a positive PCR test.
And it's not untrue that it's positive, right? Like I'm not trying to say that there isn't virus there. There very well may be a virus there. The point is though that it doesn't seem to be a virus that's enough to cause infection. And actually, if we just go through the rest of this paragraph, I mean, they're really tooting their horns about how awesome this PCR stuff is. But if you go down here, it says that between laboratories, you take the same sample, you run it between laboratories, there can be off as much as 14 cycles. And within a laboratory, you have 3 cycles of wobble. I can live with the 3 cycles of wobble, but 14 cycles between laboratories? I mean, that's kind of shameful for like really saying like, here we have a gold standard testing method. Like
within laboratories was 14 cycles, right? The interlaboratory median of CT values within, but that's within laboratories. Right? Oh, so it's ambiguous wording.
It's a horrible piece of English, but what they're trying to say is you take the same sample, you run it in 2 laboratories, they might be off by 14 cycles. And within a single laboratory, you run that sample twice and it'll be off by 3.
Which is basically in line with my relationship to Rtpcr. So one of the most difficult things about working with RNA is the fact that RNA is extraordinarily unstable. Like our bodies are constantly shedding enzymes whose job is to destroy RNA. And virions, when they're no longer living inside of cells, are not particularly stable structures. And so you have this 1-2 punch where on one hand you have the fact that you are in a world that is surrounded with these RNases, which want to digest viruses as much as possible, or digest RNA as much as possible. And you're also dealing with the fact that viruses fall apart. So if you're taking the same sample and one lab stores it a little bit less carefully than a different lab, you're just going to get degradation. You're going to get little minute changes in the way that they handle the material, and then you're going to get this massive difference on the outside, which is kind of the hallmark for a test that isn't particularly good. And yet, here we are still using or I guess we're not anymore. I feel like people have really moved towards antigen tests. But when this was written in 2021,
they seem by the conclusion sections to really still think it's the best thing since sliced bread. And that's just like, I just don't know how you can look at this data and conclude that. Like, I'm looking at their antigen tests in this paper and I'm like, sound pretty good to me. Like, there's another, look, let's go to the page 6. They concluded, blah, blah, blah.
Sorry, what? Can you tell me?
US CDC in a study with various age groups. I'm at the top of the paper. I'm sorry. Yeah. This paragraph, exactly. They concluded that the virus couldn't be cultured more than 10 days after the onset of symptoms. All right. So if you correlate that data with the idea that a certain cycle count is associated with more or less, as we just went through, right? Days of culture, right? You can't culture it. That's a certain cycle count. And you know that it's not culturable, i.e. contagious in any clinically meaningful sense after 10 days after the onset of symptoms. You could correlate those two together to come up with a meaningful cycle count where your PCR test would actually be meaningful. But it seems like it would be in the sub 30 range, right? I mean, that's in this paper. Like, I'm just saying they're not even internally consistent here. By the way, we didn't just like pick this paper out of nowhere. It's one of the most highly cited papers if you go to get a review of the testing procedures in SARS-CoV-2 at the moment. So I'm like, they have all the data they need to actually turn the PCR into an effective test, at least given what they knew at the time, not necessarily after what we know in the next paper, but still, it's just like, anyways, we can keep going.
I mean, I think that this will become more apparent when we start looking at the figures in the next paper, because it is kind of jarring to look at this conversation versus the very sort of highly quantified analysis of the other paper. So, I mean, this is, again, they conclude with Rtpcr is the gold standard for its high sensitivity and specificity. But there's problems because you cannot do this test at the point of contact. Rtpcr always requires that you take the sample, you drive it away, you do your PCR somewhere else. It's not rapid. It takes a couple hours for things to amplify. And so it's also expensive. Like an Rtpcr machine $5,000, $6,000, that one's $36,000. Like they're expensive pieces of machinery, and so if you're trying to find something that's going to work for the entire world, it is both a highly sensitive test. It is a very difficult test to run. It requires a competent technician.
There's a lot of interlab variability.
And so it's hard to get on board with it, and it's hard to understand how they've come up with a sensitivity and a specificity that is this high, unless it's in a highly controlled setting with a very, very highly trained technician.
I think that the specificity and sensitivity are reasonable reports. Like, I do think it's extremely sensitive and specific. I just don't think that matters. That's my beef with this paper, personally. Just to be like absolutely clear. Like, the fact that you're super sensitive, if you can't correlate it to what's actually happening in the patient, doesn't make it a good test for predicting what sort of actions you should take.
But I'm also adding on top of that pile, which is that the sensitivity and specificity comes down to the fact that these studies probably used a small number of people that were very highly trained, that were doing these Rtpcr tests in order to give these numbers. Because this, to me, is the side effect of somebody who really knows what they're doing, and I don't think that every single technician in the country that's
they're all using the same machine and
inside of each one of these studies, right? So it's like,
by the way, do you get a sense of how they calculate? So they say how they calculate sensitivity and specificity. Do you get a sense for how they calculate true positive versus false positive versus?
I think that they confirm with other methods. So they're looking at seropositivity. They're looking for radiology confirmation. And so a true positive is one that actually, and I think it also has to do with the fact of like developing disease as well.
Not sure about that.
I'm not sure about that. There's alternatives to classical Rtpcr tests. Like I talked about, they talk about this Sherlock assay. They talk about RT-LAMP. RT-LAMP was a really crazy one, which I wanted to show you because it was just so weird. Here, where are we?
This is like a one-step PCR thing.
This is a one-step PCR, and it's just it's a cool technique because it's very, very cheap and it's very accessible, but I've never seen anything like it. So basically what you end up doing is you take your original sample, you find a target region that's inside of your original sample, and then you start to amplify it in such a way that you create these looped sections. And because of the way that the looped sections are designed, you continuously produce more and more of these looped sections as one single long piece of DNA. So normally what you would get if you do a PCR is you get just the product of the segment that you're looking at. But here you design primers specifically so that instead of creating a high fidelity copy of what you're interested in looking at, it creates this weird kind of monstrous high molecular weight amplicon, which means that you just have one big chunk of this DNA where you get the same piece of information in it over and over and over and over again. Like, this is one repeat. This is a repeat that's in the other direction. This is another repeat. So you're basically like attaching these barbells to each other and extending them over time. And that means that what you can do is, because you have such big pieces of DNA, you can actually use a color indicator for showing how much of the DNA is there, which means that your detection for a quantifiable assay is much cheaper than the $6,000 machine. This is a sous vide machine. This is an enamel pot, and you have your reaction floating in it. And rather than having to constantly be changing your temperatures and melting the DNA and re-annealing the primers and everything else onto it, you just maintain a single temperature the entire way through, which is kind of a stunning development in terms of PCR. I know that PCR just in general is not a particularly exciting field, But the idea that you can use it in order to do this kind of really cheap garage microbiology is really interesting. And I think that this kind of technological developments, like I think that the biotech of the future is going to be a lot of really small, cheap labs working on questions that are kind of weird and esoteric and out there, rather than being exclusively focused on the super high-tech world of laboratory science. Because it's a business. Something that I get while I read these papers is really the amount of money that has been generated by the companies that produce all of these testing reagents. There's a set number of companies in the world that actually produce them. You know, it's probably maybe like 10, 15 big companies that are producing all of the plates, the pipettes, the reagents, the kits. And so it's fascinating to watch the degree to which this has ramped and benefited a relatively small number of actors. Let's see what else they have in this paper about RT-LAMP. So basically, RT-LAMP is a benefit because you get a much, much faster analysis than you would otherwise, and it doesn't have the huge infrastructure necessary for it. Then they basically start to go way out there where they're like, look, we can use CRISPR, we can use Cas13 or Cas12, which is an enzyme that's capable of cleaving this fluorescent marker as well. And so if the target sequence is there, then it can bind to it and it can cleave it and it can release light. And it's entirely possible that down the line, these things will become a legitimate alternative, but that hasn't really happened yet. These are basically just potential, right? Because it's saying that and then there's also, you're still carrying forward this fundamental assumption that they've made at the beginning of the paper, where they're like, look, if we start using RT-LAMP, which is this large amplicon PCR, in combination with CRISPR-Cas, we're going to get really, really fast detection systems, and they'll be crazy precise. We're going to be able to detect attomolar concentrations of DNA, which is in my estimation far below
10 to the minus 18th.
Like that's a concentration of DNA that is not clinically useful. And so this desire to identify every single last transcript is a technological obsession rather than a scientific one, because the scientific obsession would be to focus on, okay, well, what does one transcript do to disease? How many transcripts do you need in order to cause disease? And what is the disease that these transcripts cause? And then go back and be like, okay, well, we can quantify disease by this proxy. And
it's probably different for different people in different boats, right? And that's really important too from a scientific perspective. I mean, science is all about figuring out what's happening. Not necessarily just pinning the murder on somebody.
Yeah. We talked about this, like Sherlock, which is again this CRISPR-Cas based thing. They basically come up with a collection of very advanced methods for being able to detect this that are not exactly PCR or Rtpcr, but they're all based on the fact that you have a small amount of starting nucleotide sequence, and then you gradually amplify it, and as you amplify it, you end up producing vast quantities of the stuff. And that way you can detect it. Antigen analysis is fundamentally different because antigen analysis does not amplify your sample. We don't actually have a good way of being able to amplify proteins. I stand by that. Like, you can't even sequence proteins. Like, you can sequence DNA, you can sequence RNA, you can amplify both RNA and DNA, but if you've just isolated the protein, it's kind of a dead end. You can't chop it up to figure out what its sequence is. You can't get back to the original DNA sequence from it because it's degenerate, which means that you'll have multiple codons at each position. You just kind of have this piece of protein, but you can test. And the tests that they're using for these, I have some slides. The tests that they're using for these have a couple of different forms, and I want to go through them because they'll be useful later. So one of them is ELISA. And now that we have kind of talked about antibodies a little bit, it's pretty easy to explain ELISA. They're often done in plates like this. And the way that it works is that you attach something to the bottom of the wells, and then you detect what is on the bottom of the wells and what sticks to it. And the way that you attach it can be multiple different things, right? You can take your sample, you can add it to the wells, you can force it to polymerize with the plastic, then you can add antibody that binds specifically to just your antigen. This is this one pink one in a sea of gray. And then you have an antibody that's there that has a little motif on the back of it. And they literally just add this piece to the butt, and it's able to take a chemical that is colorless and make it colorful. And so what you see here is that the wells that have a more intense color are the ones that have more of these molecules that have bound to the bottom surface of the plate. It's a pretty straightforward method. It is...
Then you just shine a light through and see how much of the light is blocked at the end of the day for your output.
And so it is a hugely popular method for detection, but it's expensive.
They sell these kits. It's kind of like baking a Betty Crocker cake or something.
Yeah. How expensive do you think an ELISA kit is?
A few $100s. I had a job one time where I had to run these kits, well, amongst other things, and I kind of always enjoyed the days, the ELISA days, because it was like, you know, you're basically just baking a cake from instructions. You got all of your little reactions. They come in this box. It's like add solution A to this thing, you know. Put 3 drops of this in this well. They're kind of hard to mess up, which is cool in a way, actually, if you're talking about standardizing treatment, except it doesn't seem like everybody's using the same kits everywhere. It seems like there's something like 800 different kits of these available that are in use right now. They probably all have wildly different specificity.
And so when you pull up one of these kits, you get this really shockingly perplexed graph, right? Which is, do you want avian, bovine, camel, canine, chicken, donkey, duck, equine, feline, whatever? You have all of these different animal choices, and you're like, what does that mean? And what that comes down to is if you go back and you look at this ELISA picture here. Oh my goodness. If you look at this ELISA picture here, what you can see is that sometimes you have multiple antibodies that are involved in the experiment, where you have this primary antibody that's complexed to the bottom, and then you have another antibody that comes around and detects it. So this is the example down here, right? Where you have an antibody that's conjugated, then you have another antibody that comes along and is the desired antigen for the complexed antibody. And here you would have, let's say, a chicken anti-goat antibody, and this would be a goat antibody. And so that is why when you go and you look at these ELISA kits, they're basically like, well, what animal would you like? Because they've created antibodies inside of each of these creatures that are specific to one another, and that's what's necessary for actually running the test.
I mean, it saves you a lot of money at the end of the day because you only need like a green antibody as your final antibody, right?
Yes.
And the green one's made out of some weird species like a donkey or something. And or your second, then you can basically have an anti-donkey green thing. And so anytime you have a specific donkey antibody, you can tag it green.
Right.
Save yourself a lot of money because these fluorescently tagged antibodies cost hundreds of dollars for like a little tiny vial of them.
And this, like, you know, you basically go on here and it's just whatever you can think of, you can buy an ELISA kit for it. They're $300 to $500. The ones that are more obscure, difficult to source antibodies for are going to be more expensive. But this is just a very standard method of measuring the amount of antigen that's present, because it gives you this very clear readout. It allows you to do multiple replicates. It's very much intended for you to be able to do high throughput screening of very many tests. Lateral flow antigen tests are really interesting to me because these are the ones that are like pretty robustly functional, and they operate on a similar fluorescent color changing mechanism.
It can be done in the field.
Where you basically have this this plate.
Flow cell?
Flow cell, yeah. I mean, they're usually like, this is the same thing as like a pregnancy test is, right? So you put your biological sample down on the left side of your flow cell, and it's wicked across the surface. Capillary flow brings it to this pad that's down at the bottom. And what it does is it gets first to this conjugated space. And this conjugated space contains antibody that is going to bind to whatever substance is inside your analyte, right? So if you're looking for spike protein, this drop would be full of spike protein. These antibodies would be anti-spike protein antibodies. And so when this mixes together, you have these fluorescently tagged or color tagged antibodies bind to your spike protein. And then they go over here. And what you have is you have a sandwich ELISA, right? So if we were to go here, this is what the sandwich ELISA looks like. You have one antibody that's bound to an antigen. And then you have another antibody that senses that entire complex. And so the sandwich ELISA basically aligns those colorful positive antigen carrying antibodies. And then you have a second line that is your valid control, which says like, yeah, we're just detecting human antibodies here, and here they are. And so everything is present. And these are very, very effective because of the shape specificity and because of the way that if you have lots of spike protein in your blood, or you have lots of IgG in your body, that is a very accurate measure of whether or not you've actually been exposed to this disease. In terms of the rest of the stuff on this paper, I think we should move on to the Nature paper. What do you think? Do you have anything that you want to add here? You're looking at some kind of graph.
I wish I had a way to just throw this paper over there.
If you dropped it in a slide in the slideshow, we can get it. Right. One of the tests that they propose is lung images, right? Which is a little bit farther down.
It's here. About that. They had some cool AI deep learning ideas that were kind of nifty.
Right, where you should be able to. So, like, you have a severely impacted lung is very congested. It has a lot of these fibrous plaques in it. We've talked about the way that they're formed during the course of the infection. And it's not a particularly specific test in the absence of some kind of molecular test, because lots of different things could be causing some kind of ground glass infiltrates, which is everything from viral pneumonia to tuberculosis to COVID. And so, and imaging is crazy expensive. So that seems a pie in the sky idea.
They're basically like, look, the real gold standard would be if we could just look at people's chest x-rays and see if they have pneumonia or not. Because the disease is actually defined by the pneumonia, ultimately. At the end of the day, the extreme disease, sorry. The one that's killing people.
All right, what are we looking at?
We don't have enough technicians to do that.
What are we looking at here?
Oh, so this is just kind of, I just want to reiterate that it seems like the way the PCR test could have been implemented in a way that would have been useful is, A, if they could correlate the severity of infection to cycle count. And in lieu of that, they were able to do it based on whether they were able to culture the viruses after a certain point. This paper came out pretty early in 2020, and they were looking at a little cohort of patients and tracking their cycle counts over time. And what's really interesting is that nobody who was sick had a cycle count above 30.
Oh, which figure should we go to?
Go down to there's one central figure. Keep going. There you go. So everybody who got sick was at least below 30. And most of them, well, let's not say most of them. Just say everybody was below 30.
So this is 10 cases. And so what are we looking at here? Walk us through it.
Just cycle. I don't really want to point out anything other than we're looking at cycle count over days. Okay, so the person, you only see two dots.
Cycle threshold or cycle count?
Cycle count. I mean, isn't that threshold? This is the number of counts, right? It's how many cycles.
A cycle count is the number of times that they run the PCR test. Cycle threshold is the time at which they say that it is positive.
Okay,
So like,
this isn't about positivity at all. This is just about the number of counts. How many cycles there were
in the PCR test, or
right.
Before it was considered positive.
Before you got a product amplified.
Okay, I see. So it's cycle threshold. So the threshold cycle value is over
before
here.
It takes off, right?
Okay, and then we have 10 cases.
Not a single one of the cases was over 30. This paper actually goes on to argue that a lot of these patients that don't drop down to 45 ever had completely recovered and were discharged from the hospital.
So what you're saying is that is this a time course?
Yes.
I see.
If you go down, you'll see days. This is
days. This is what a graph. No, there's no more to this figure.
Oh, really?
Yeah. So basically, you have days here. From day 0 is days since symptom onset.
Okay.
Day 21 is over here on the right. And so what you're looking at is that they tested these people using PCR on multiple times over the course of the progression of the disease. And at no point in the progression of the disease did any of these 10 people who were experiencing symptoms have a cycle threshold value that is above 30. And so the point here is that that would suggest, since all of these cases resolved over time, that if you set your threshold case to being above 30, that you'll be catching all of these people on their recovery down to baseline. And everybody recovers down to baseline.
People. That's what they're saying.
You will catch sick people. You'll just also catch all of these other people that aren't sick in the moment.
Sorry, my bad. That's exactly right. They actually go on here. This is maybe interesting, but not totally what we're talking about. Half of these patients who still have their CTs above 30 at the end are no longer sick and are discharged from the hospital. So their final conclusion, if you zip down to all the way to the bottom of the paper and you read the last paragraph of it,
we conclude that CT values of real-time Rtpcr assays decrease as the patients recover, while some of them remain positive, although the patients were asymptomatic. Positive results of Rtpcr for SARS-CoV-2 might not always correlate with the degree of infectiousness. The discharge criteria by Rtpcr testing might have contributed to a COVID-19 patient's long-term hospitalization and should be reconsidered. They're basically like, look, you guys, if we are using this as the baseline for saying whether or not somebody can leave the hospital, it's going to create insane problems because we're not going to be able to treat disease linearly with cycles.
And we're going to have our hospitals full of people that aren't sick.
And I think that this is a great time for us to go to our other paper because it is a wild ride. This one I was obviously much more enthusiastic about than I was about the methods paper. You can always tell my enthusiasm for a paper based off of how much I highlight. So
hopefully everybody's with us now. We have to go through the methods first before we can reasonably talk about using them.
Exactly. And so, okay. The question is, does your amount of SARS-CoV-2 lead to a correlated illness, or does it not? And so this is a study that seeks to try that. This establishes a novel SARS-CoV-2 human challenge model, which means that what it's doing is it is actually infecting people with SARS-CoV-2 who have volunteered. 36 volunteers were inoculated with 10 CID50 right off the bat. 10 CID50. This is a tissue culture infectious dose. And so they were given 10 times the amount of virus that you need in order to infect cells in a plate. And then this wasn't randomized. They basically knew what was happening to them. It was totally open, but they squirted it in their nose and they were like, we're going to put you in a containment unit and watch you.
So right off the bat, I do think this is a fascinating study, but we see a serious problem with this study design immediately. If this is more of a discussion, I would love to hear if somebody... Actually, does anybody want to chip in really quickly and mention the study design problem here that's kind of obvious? Anybody?
Like, what do you need in a
This is
study?
driving me absolutely nuts when I read this, because I love this study, but I'm just like, guys, come on, how could you not do this? Anybody?
Like if you're basically saying that we're going to take some stuff from one person and we're going to put it on another person, what would you need in that test to be able to say that the transfer that you've made is actually causing the effect that you see?
What do we say for... You guys are all designing experiments, theoretical experiments right now for your final exam. We're always like beating you with this, like, you have to... You got to control your experiment, right? Sorry, I just didn't want to wait anymore. They didn't take the sputum from uninfected people or from non-viral people and put it onto an additional 36 people, which would have been really fascinating. Because my first thought is like, man, if you just take a healthy person's snot and stick it in my nose, I might have a pretty crazy immune reaction to it. I might. And I would have loved to have seen that data as we go along. It doesn't necessarily take away from any of the rest of the stuff, but it is sloppy, and I was a little bummed out about that.
I mean, you want to be able to know clearly that... I think that anytime that you're exchanging biological fluid between individuals, it's hard to imagine that there's not going to be some kind of reaction. And so you would want to see... You would want to see it run the other way, but I can also understand I think that it's just not in the style to do it. I don't know. I have no idea. The primary objective of the study, and I think that this might be partially why they're doing this, is because they're viewing this as being an incremental step towards doing a larger and larger study. Because if people remain focused on COVID to the degree that they have been for the last few years, then it makes perfect sense that what we're going to start doing is we're going to start doing more and more challenge trials. And so they're like, the primary objective of this study was not to do any kind of clinical controlled trial where we really get to the bottom of this. It was literally just to find an inoculum dose that is going to give 50% or more participants symptoms.
I mean, they excused themselves nicely from that. I appreciate
But if that was the point of the objective, I mean, like...
Well, they spend a great deal of the discussion making light of the other findings from the study. You know...
Like, the secondary objectives were to assess virus and symptom kinetics during infection. And this is where...
Well, that's an underpowered conclusion without a control.
Yes. And so that's where you really... The primary objective doesn't really need a control. The secondary objective is where you really do start to get into a weird thing, because it's like, people carry all kinds of viruses. There's all kinds of things that you could be transferring in the sputum that are leading to problems that don't necessarily have to do exclusively with COVID-19. And with something that is so weird, right? Like, we've looked at this and we've seen that you have a sickness and you can apply all of these different drugs to it, and almost none of them show massive improvement. And
or even discernible improvement. Aside from the steroids.
Aside from steroids that just knock down your immune system.
It wasn't like a amazing effect. It was like a potential correlation.
No, it was a good effect. It was the only one. The corticosteroids were the only ones that were like, this will fix people.
I have to look it up. It wasn't like a strong adjective.
My point is that,
because the corticosteroids lead to other problems is the thing. People get secondary infections and stuff. So it's not a silver bullet.
Yeah. But the point is that the paper displays a kind of shocking lack of intellectual curiosity, where it is taken for granted that doing this transfer is not in and of itself interesting. And I kind of find that to be the case in a lot of science papers. I remember when I was working in grad school, we were looking at these like bacterial colonies grown on agar plates. And I would always be captivated by these questions that seemed to be fundamental questions about the method. I was like, well, what happens to the bacteria when you culture them in a laboratory for an extended period of time versus culturing them somewhere outside? And that just wasn't the question that was worth asking, because it's not a question that gets you farther down the line towards a piece of technology. It's a question that slows you down a lot. It's a question that kind of allows you to navel gaze a little bit, to shoe gaze, to kind of be like, well, are we even doing the right thing? And that's not a question that science in its current iteration is that interested in asking. And so they wanted to assess these secondary objectives, which is virus and symptom kinetics during infection. They take for granted that they don't need to control for taking a healthy volunteer and having a group where they put the healthy snot in somebody else's nose. They did succeed by their own assumption of hitting all primary and secondary objectives. So of their 36 participants, every one of which who got inoculated with COVID positive snot sputum, only 18 became infected. So that's 53%. We'll talk about viral load dynamics a little bit later. There's a difference in nose versus throat. No adverse events in this population. Presumably they selected them very carefully.
They did. They speak to the fact that they picked from 25,000 volunteers to pick the toughest, most healthiest, least likely to get them sued people.
So mild to moderate symptoms were reported by 16 of the participants, which began about 2 days after they got inoculated with this bolus of virus. And 2 of the participants remained asymptomatic, which means that they had positive test results but no symptoms. The rest of the 47% didn't develop infection at all. So they weren't positive, they weren't showing any kinds of symptoms. They just took it and moved on with their lives, which is really interesting because you wouldn't expect that. On the surface of it, when you are talking about taking a sample of something that is supposed to cause disease and you inject and you put it onto people's mucous membranes, you would not expect that almost 50% of the people wouldn't develop disease. So this is, I don't know, it's just so cool. It's like a real change in our understanding of what viruses are. And it's obviously a small trial. It's obviously in a very specific group of people, but it's still very, very interesting.
It's interesting that statement of 2 participants remaining asymptomatic is in direct contradiction with the table 1, which says that 17 patients were symptomatic of the infected people.
Well, maybe they didn't have maybe the symptomatic people, there were 18 people who were symptomatic, but...
No, these are 18 of the people that were infected. Total.
So uninfected sero...
These are the people that were excluded from the study because they were seropositive before it started. There's actually 34 people in the study.
So they're basically missing 1 person from that analysis, is what you're saying?
No, I'm saying that there's only 1 asymptomatic person.
But they say that there's 2.
I know, but they don't in the table.
Well, so there's some number is missing somewhere. We'll look at that in a second. They did speak about asymptomatic stuff in this kind of like glossy way, where it was hard for me to follow in places what it was exactly that they were trying to say. But the point that I want to make before we move on from this is that no quantitative correlation was noted between viral load and symptoms.
Yeah, that's the big pink bold finding.
With high viral loads present even in asymptomatic infection. But 50% of people don't get infected.
So next time there's a pandemic and you guys are all the scientists developing the tests and stuff, you need to come up with a test that actually predicts outcomes. That is the actual real life gold standard. Not whether you can...
Go ahead, sorry.
What is it?
Well, what I was saying here is that like no quantitative correlation was between viral load and symptoms. What they're saying is that there was somebody who didn't experience any symptoms, but they still did have a high viral load, which would make them somebody that you would want to quarantine because they're a spreader.
But there's also 33% of the people who weren't infected had symptoms.
Right. And so that I think is together is just this wild ride that we're about to go on. and a wild ride of exploration. Okay, so again, I have started to kind of glaze over the introduction paragraph. Was there something that you seem like you have things highlighted there? Is there something that is worth?
Not really. We're just talking about the experimental design for the most part. I don't know why they waste a whole page on this drawing. It's kind of boring.
Okay, so 36 healthy volunteers, 18 to 29, enrolled according to this defined inclusion. They screened them basically for everything. They were like, we are going to make sure that nobody has comorbidities, nobody has any kind of issues with kidney or liver function, all their clotting and blood serology looks good. They breathe really well, their hearts are really good, their chests are clear. We have picked well.
I was also like, it seems like over here, I don't know, I was having a hard time making sense of this, but it seems like they gave the first 10 people a prophylactic dose of remdesivir.
So they ran this multiple times. And
okay. So that's not the data
No, this isn't the data that they're talking about. They're basically like, okay, so the first time that we did this, we were really freaked out that people were going to get so sick that we were going to kill them, that we gave them a prophylactic dose of remdesivir.
I see, that's not a different...
right, so the initial cohort was 3 sentinel individuals, and then 7 extra people, so 10 people. These first 10 people were given remdesivir for as soon as they found any 2 swabs that showed that they started to get SARS-CoV-2, because they wanted to make sure that they would be able to stabilize them. And these are antiviral. This is an antiviral medicine. But they basically they took the data to the review board. The review board was like, oh, this seems like it's kind of not necessary. Then they got the clearance to do a larger study. They did a small cohort of 3 and then tried it again. And then they did the rest of the people. So they had, 28. They had like a bigger study of 28 people. So once they stopped using this preemptive remdesivir, they were then able to set a higher criteria where they were like, okay, so we're going to wait until people start to get really sick, and then we're going to give them monoclonal antibody therapy, which we know works by basically coating the virions and helping the immune system of the body be able to collect them and remove them. And they were like, but that was never required because nobody ever made it to being that sick, which means that they're choosing their people carefully, right? Because people that have anything wrong with them on that count are going to give very different results. Then the question is that you have a choice here. You're like, how much virus are we going to give people? Because the primary endpoint is to show that we can find a dose for this experiment where at least 50% of the population that we're testing is going to get infected. And they choose 10x this tissue culture dose. Tissue culture is when you have just a monolayer of cells and you add virus, you squirt virus on top of it.
It's interesting because it would have been so cool to have done this study with, they had 26,000 volunteers. If they did it with several cohorts and they used a different TCID50 for each group, that would have been able to answer the question a lot better, it seems like. As opposed to being like, well, our target was to get half the people infected.
I'm sorry, say that again. I zoned out. I'm sorry. I was looking at the paper. Tell me again.
Well, they should have done it with like several groups of 30 whatever people, and they should have done it at different TCID50s. And then they would actually be able to answer their question, what's the minimum amount necessary to infect half the people?
Right, exactly. And so like if you continue to drop your TCID50 lower and lower and you're still infecting 50% of people, that's a pretty clear marker for you that there's something about the procedure itself that is worth looking at. All right, so the dynamics. You have 18 infected individuals and you're looking at virus shedding by Qpcr. And we just talked about Qpcr. And so it becomes quantifiable in throat swabs at 40 hours after inoculation. There's a bunch of details here, but what I really want to get is...
That was the table, by the way, that I was referring to.
Can we come back to it in just a second? what I really want to get was this FFA versus Qpcr. So I wanted to show this chart here. So FFA is a focus forming assay. And this is actually a question of functional virus. So Qpcr is telling you about total number of DNA copies or RNA copies that are around, but a focus forming assay is one where you do dilutions of your sample in order to be able to count how many viruses are actually in that sample. And so on this plate over here on the right, you have a dilution series, and you go from wells that are completely clear to wells that are basically completely dark. And each one of these little light spots on these dark wells, you can see that there's more of these light spots here, is a viral plaque. And so what that is that the virus is, the virions are added to some tissue culture well, and they float around and they latch on to a cell. They get into the cell, they kill the cell basically floats away and leaves a blank spot in the place where it was. And because of the dynamics of viral spread, the viruses or the virions aren't able to like float away. They can't move. They'll always kill locally. And so you'll get this plaque that forms. And so it's obvious that you can tell that viruses are still infecting these cells by virtue of the fact that you have these plaques. But if you look at the comparison between PCR and this plaque forming functional assay, what you see is that the PCR was positive for 28 days. So they say, so at day 28 of inoculation, 6 of 18 participants remained PCR positive. However, if you're looking at this functional assay, you see that at 28 days, you don't have anybody who's positive. In fact, you're mostly clearing people at 10 days. And so it's kind of just another piece of evidence in the pile that says that if you're looking at Qpcr alone, you're really overestimating the amount of active virus that's around. And that's bad news given what we do. Like the way
the amount of viable, like infectious virus.
Yes, exactly.
It's like all about it's all about outcomes.
It's all about outcomes.
But for some reason that has not been the standard, and it's very bizarre.
So what did you want to say about this table?
I just was looking at. I just thought this was interesting. But the symptomatic. It looks here to me like 17 out of 18 of the infected people were symptomatic, and 5 out of 16 of the uninfected individuals were symptomatic on at least 2 consecutive days. Some really good markers for whether you've been infected or not seem to be A, whether you have symptoms, B, whether you've had a fever, because nobody in the uninfected group had a fever. So if you really want to know whether you should wear that mask on the airplane or not, seems like having a fever is a good sign. You should definitely wear that mask.
In general, if you have a fever, wear a mask when you go outside.
I mean, it might seem obvious, but like that's a huge finding from this paper. Like 7-0 is a huge finding. Like, this is also a huge finding too. The antigen tests. Is this
antibody titers?
Are these antibodies or are these antigens? Neutralizing antibody. Okay, so this is like seropositivity, and that's I mean, it's undetectable versus like in the hundreds of detection units, whatever these are.
Thousands for the other one.
Yeah, so these are great findings actually. So you got a fever and your blood tests seropositive in that moment. I mean, could be that your blood tests positive later on too, so I don't know the answer to that.
And you could have a fever later, like you could be you could be like one day before, right?
Right. But if you have a fever, or if you have a fever, you should probably wear the mask. Like
the next version of this paper would have to actually look to see the sort of the dynamics of infection in the early days right after exposure to see if you're actually capable of like carrying that to somebody else. And you could do that in animals in some way. You could do that in people if people were willing. But it's definitely
like this is also interesting too. These two folks over here in the far right hand column. If you hold on.
But they seroconverted, right?
Exactly. So they had already experienced it months before, let's say.
Mm.
Their antibody levels are like
no, not months before. They seroconverted between screening and acceptance and start of the trial.
How long do you think that was? Probably a couple months.
I figured it would be weeks.
At any rate, like their neutralizing antibody count went down significantly compared to these people who are in recent infection, right? So that would be an interesting study too.
To see like how quickly the dynamics of these things recover, where it's like if you're testing somebody a year out, would you still have it? What about 2 years out? What about 3 years out? But we'll only ever really know the answers to these questions as long as there is money to be had studying them. And I don't know how much longer that's going to hold out, because it seems like the arc of COVID mania is starting to close. And, science, this stuff's expensive.
Well, the best thing we can do is just learn and act smarter next time, really.
Mm-hmm. So the summary is, despite relatively high levels of late Qpcr detection, which is in line with what the previous paper said, that like Qpcr is the gold standard for detection, the latest that viable virus could be detected was day 12 after inoculation in the nose of one participant, and day 11 in the throats of 2 participants. So now we're looking at figure 2G. And this is also really interesting to me because they purport that this data is significantly different.
That was...
There's a bunch of this stuff in the paper where you look at the spread of the data and they're telling you, you're looking at a picture of a Rorschach test and they're like, this is a painting of a tulip. And you're like, I don't know about that. And this is one of the first places where I noticed it, where I'm like, I don't really think that these days are particularly different. And they also do something that's stunningly dishonest, which is that they change the axis of these graphs. In theory, you want to compare figure G and figure H here, right? Because what are we looking at? We are looking at days to last virus detection, days to first undetectable, which is the same thing. They've just written it differently. And this is by this focus assay. So this is the in vitro infectivity assay. This is the Qpcr. This graph goes up to 17. This graph goes up to 14. And so what this is showing you is that these two groups are actually located somewhere up here, and they're even going up beyond the margins of this graph. And so it makes it hard to see visually and clearly that PCR is extending positivity by several days past where it should be.
Yeah, if you go to the top of this, I had another similar complaint. In this first panel A, they're making an apples to oranges comparison between viral load by culture and viral load with Qpcr with the same axis on the left, which seems just completely preposterous to me. Like, you're not, like, colony forming units and how much virus you have by Qpcr, copies per milliliter. It's like, those are not equivalent units. Those do not belong on the same graph at all. The trend is actually quite overlapping and quite interesting, actually, with this tail, especially if this tail is significant.
Yeah, because I mean, like, I think that it's actually quite informative to show it this way, because it's showing you that basically on day 12, you're still getting some amount of viral load, but you no longer have any viral load by this focus assay. And you're showing that if you're testing from the throat, you are getting...
But copies of the genome per milliliter based on, like, you don't get copies of the genome as a direct readout of PCR.
You do. You get not copies of the genome. You get copies of your amplicon. That's what the control. That's why it's Qpcr, right? Because it's actually giving you copies of virus that you start with and that you end with, because you're using your controlled series dilution, and your cycle threshold is telling you how many copies of this you actually have before it becomes fluorescent.
How can you compare that to what you have happen in a tissue culture, though? Is what I'm trying to say.
All that they're trying to show here is that they're trying to show that the PCR positivity extends far beyond your functional
then you
assay
should normalize
positivity.
those two curves. This should be normalized to this curve. The max should be the max.
Yeah, that's probably true. It's definitely like they're definitely not linear relationships, and so it's kind of, you can already tell right off the bat that they're doing these sorts of, they're doing kind of weird stuff with the data. Let's see. And again, here, right, they're trying to say that this group of first quantifiable virus is significantly different from the nose versus throat in both of these cases. They're saying that this is very different. And while the distributions do look different, they overlap deeply. And so to come away from this and to be like, this difference is massively significant, because that's what these two asterisks mean, right? It's usually that you have a p-value of like 0.001 or something.
Yeah, those are nice spreads.
Yeah, these are nice spreads. I'll take that. But I just...
Yeah, I think that the word that people use in the literature a lot is, if it's less than 0.05, it's... sorry, if the difference is more than 0.05, it's significant.
At the p-value, you mean?
Yeah, the p-value, which is like a standard comparison of spread between two groups.
Mm-hmm.
But as you can see, something being quote-unquote statistically significant and satisfying that p-value, as in figure G over here, is a very different matter than something like panel E over here, which is also statistically significant by the same test, but they're very different situations. So whenever you see statistical reportings and p-values, it's really important to actually see the spread of the data yourself, because things that are quote-unquote statistically significant are not all the same.
Right, because, I mean, even, so if we look at legend for E, man, this is wild. So 4 asterisks is a p-value of 0.0001.
So I said greater than 0.05, less than 0.05.
So what this is saying is that the p-value between these two clusters is highly significant.
And what that means is there's a less than 0.05% chance that the effect is due to random occurrences.
But because you have such a small sample size, the idea that you could get such precise estimates of probability or error is just not really reliable. And so you have to be very careful when people start reporting such massive significance. Like, you can't publish in Nature without reporting massive significance, but to say that this is massively, like, if you were comparing the FFA versus the Qpcr and you told me that they were massively significantly different, I'm totally with you. But if you're comparing in between these two groups, looking at the spread of the data, I'm like, that's within the threshold of noise, and you're doing something crazy to your numbers to make it look like that.
I said that wrong too. I need to correct myself again.
Tell me.
If your p-value is less than 0.05, that means that there's a 5% chance that the null hypothesis is correct, so that the effect is just due to random chance. If it's 0.001, it's less than 1 in 1,000. That doesn't change anything.
And it's but this is 0.0001, so
right,
it's less than 1 in 10,000.
Right, exactly.
And for you to tell me that there's less than a 1 in 10,000 chance that these are spread like this, on accident, I don't know. It's frustrating because you want to see really good data on this and you don't see it here.
You're sure they're not comparing this group to this group?
Why would you?
That looks like 1 in 10,000.
Yeah, exactly. They're not. Because you would only have one p-value here if you did. So basically, I'm not a fan of that graph. They are also looking at the levels of neutralizing antibodies inside of people as they are infected. And they see that everybody has kind of this very similar trend where they pretty much get to a high level at day what is it, day 14, and then they plateau to day 28. There's one person in the uninfected group who seroconverted between day 14 and day 28, and that's the person who they ended up having to throw out of the trial, as far as I understand it. I think that's this person who's in... No, that's not that person. So they basically got to this guy, and they were just like, no, we're not going to have you. This
is kind of interesting.
Mm-hmm.
Upper left.
Okay, so UP-SIT change. The UP-SIT is a University of Pennsylvania smell identification test. And the way that the smell identification test works is that, and this is funny, they give you a series of scratch and sniff cards. And they're basically like, what does this smell like? And you can see here that there's a little scratch and sniff card here. And then it says, what does this smell like? Gasoline, pizza, peanuts, or lilac. And so when you're looking at these results, they're basically running people through this gamut of tests. And they're like, how many can you actually accurately identify? And what's funny to me is that a lot of the people don't seem to have the ability to really piece these smells apart very well in the first place.
Yeah, I was gonna. Where's the? Did they look at did they give the uninfected people this test also?
I'm not sure. I don't think so.
I'm trying to find that.
Anosmia.
Yeah, nice. They did. So it seems like it's actually smell disturbance is actually a really nice predictor, because only one person in the uninfected group at day 28 showed that.
Exactly.
So it seems like that would actually be a better test than PCR.
I mean, we haven't quite figured out our false positive rate for PCR. Dr. DeLay really has it out for PCR today.
I'm just
folks.
saying, it looks like smell tests are a banger. Like, it looks like it perfectly predicts the amount of whether people are infected or not.
It doesn't predict with a time sensitivity. And that's the thing. Like, the PCR test could tell you before you get on the airplane if you haven't lost your sense of smell yet. And there's no sense in how it overlaps.
What will it tell me?
If you have really high viral loads before you've started to lose your sense of smell. That's it.
But the high viral loads won't correlate to whether I'm sick or not.
No, the high viral loads will only correspond to whether or not you're a danger to others. Doesn't matter about you.
Why?
Because the high viral loads means that you're able to spread the virus.
That's not clear from this. Why is that clear? How did you get that?
Because as you have higher quantities of virus, then you are able to spread it to other people.
But why? They only did one concentration of virus.
Oh, you're saying that because there's no titration of the viral loads? Yeah. I mean, it definitely warrants to be tested. Occam's razor says that the amount of virus that you have is going to be directly, like, the TCID 50 is related to viral loads. This actually brings us to a really interesting thing that I also have in the notes, which is that they are looking at, so TCID 50 and droplets. The TCID 50 is basically like a marker of how many viruses that you need to actually get in order to get infected. So when they did this in tissue culture, and I pulled the paper of like how infectious this is, they did a titration, and they're like, so you're in tissue culture, you have the median value of virions that's necessary to cause an infection is 1780 of these virions. So they're infecting people with 10 times that. So they're giving them 17,000 virions per mil of inoculum. But there's something weird about this, because if this directly relates to viral transmission, we talked a lot about droplets at the beginning of the pandemic, and everybody was like, is the virus aerosolized? Where does it carry? We can see that you have to give a lot of virions to cell culture, even cell culture, which is defenseless. It has no immune system. It is not able to protect itself against what's coming. You have to give it almost 2,000 virions. But then you look at the study of airborne droplets and how many virions they're thought to contain, they think that a 50 micrometer droplet contains at least virion only 37% of the time.
One virion.
One virion. But we're looking at this TCID 50, which says that you need to have almost 2,000 virions in order to, or anywhere between 200 and 2,000 in order to actually get a functional infection of tissue culture. And so this is kind of, I think, what you're talking
They're
about.
using 10 times that dose.
Right. And so I think that this is what you're talking about, where you're like, the study doesn't actually correlate to the fact that somebody that has a high viral load is infectious, because there
or
does...
that PCR reflects whether they have a high viral load or not.
PCR does reflect
no,
that they
it
have
doesn't. That's what this
How
is saying.
come?
Look, they're saying that you are only actually able to culture virus from somebody in the first few days, up to 10 or 12. This is from the last paper, too, that we just looked at. However, you can remain PCR positive for quite a long time.
Sure, but this figure
So
that...
therefore, being PCR positive doesn't mean that you're able to have culturable virus.
No, but it does correlate. So, like, depending on how PCR positive you are...
However, the anosmia is tightly correlated with the infectious activity window compared to a PCR.
Kind of, because some people are losing their smell for like a year later.
That's not what this data shows, though.
Oh, it does. They mentioned in the paper.
But there's maybe like one person. Go back to that.
Oh, hold on one second.
Go to the anosmia figure. It's like that guy.
Yeah, there's
like
that guy.
The rest of them are pretty tight, like in the first few days of the infection. It peaks out at day 10, which is also like when basically infectivity seems, at least tissue culture infectivity drops to nothing.
I mean, the peak, like, this is the thing. These things are really poorly correlated.
In the last paper, they said.
But PCR also correlates to that same peak. You start to decline. And so if you take a time course in somebody of PCR, you should be able to see where the peak is, and then be able to see that as they start to decrease in their viral loads don't correspond to infectivity, right? So like this peak, this is what they're trying to show here, that the peak of the viral load corresponds to the peak of the plaque forming assay. And so they're like, your PCR is most positive at the same time that you are most likely to infect in a viral load, whether you're taking it from the nose or if you're taking it from the throat. And then your anosmia, you're totally right. It overlaps. It's actually a couple of days later.
That doesn't...
Yeah, go ahead. So there's the anosmia peaks actually a little bit later. Like, I would have loved to see this same graph on top of the graph of the focus forming assays and the PCR assays, right? Because that would actually be a really interesting way to see how the shift in anosmia, I think, is a few days later than the shift in your peak PCR levels and your peak infectivity. But what they're showing over here on the right is that these are all of the different participants in terms of their symptoms. Most common ones were a runny nose, nasal stuffiness, sneezing. Nobody had the really severe symptoms, which were tightness, shortness of breath, wheezing, or blisters. Blisters was one of the weird epithelial manifestations of COVID-19 when it started to attack the skin, which is crazy. And all of the symptoms, except for headache, stuffy nose, and sneezing, and runny nose, were bothersome, but most of them were just noticeable or not there at all. And you can see that they also have this kind of distribution of how this is panning out. And so the worst symptoms are on day 8 and 9 after inoculation. So you have 6, 7, 8, 9. These are peak days. 8, 9, 10 are peak days. And so you see that 8, 9, and 10 are actually just before the peak of the anosmia. But then if you go back to figure 1, 8, 9, and 10 is just after the peak of infectivity and the peak of viral load by PCR. And so the symptoms seem to peak just shortly after the tests are able to say that, like, hey, you have resolved your situation.
Because there's this statement in the paper. It's like, viable virus was detectable by FFA for a more limited duration.
It was, yeah.
6.5 days in the nose and 6.25 days. That doesn't correspond to this graph at all.
It just says it's figure 2F is where they're looking.
Yeah, but what about figure 2A? Is this 2? Yeah.
So what Dr. DeLay is saying is that if you go and you look at the test, it says a viable virus was detectable by FFA for just 6.5 days in the nose and 6.25 days in the throat. And that is the data that they're presenting here. But that's a median. That's completely different from the data that's here, actually.
I'm saying, yeah. And different from the conclusions they draw, which is...
I hadn't even noticed that, right? Because so check it out. Basically, what they're saying here, look, is that they're saying viable virus was detected by focal assay for a more limited duration, and they're saying refer to 2F. 156 hours. How many days is 156 hours? 156 hours is 10 days. It's 10 days. 6.25 is not the number of days. It's more. I think that's the spread. I think that's the confidence interval.
Spread, I see.
So that is actually correct. Sorry. I'm satisfied with that. Okay, so we can keep going. This was really interesting. So symptom score versus viral load by Qpcr. There's no correlation. Like, this is just noise. And so they look at it versus Qpcr, and they look at it versus this functional assay, and neither one of these assays is actually capable of coming up with a linear relationship between the severity of symptoms and the amount of virus that is detected. And for me, this is the absolute weirdest figure in the entire paper. How can this be the case? And that's a rhetorical question, because there's not anyone who can give the answer to this. This is just a deep mystery of what it is that we're studying here, which is the fact that there is a huge difference in viral load, but there is not a big difference in symptom score. It's all over the map. And again, they do some really weird stuff here where they're like, the days to peak symptoms are significantly different than the days to peak viral load by this focal assay, but I don't buy that this is really different after having looked at a lot of data. Like, it's a little different, but not to the degree of significance that they want to make it. The complete loss of anosmia was in these 9 patients, but most improved noticeably before day 28. But 11 participants had weird smells until day 28. And then by day 180, it was only 5. So that means that 5 of the people that got sick, half a year later, 6 months later, still had smell issues. And so they basically were like, one of them had severe issues with smell. 2 of them had porosmia, which is that you can't correctly identify smells anymore. Like, you eat something that's supposed to smell like garlic, and you think that it smells like, I don't know, gasoline. And the way that people fix this is that they say, this is really interesting. 6 participants, including the 5 who had prolonged smell disturbance, received smell training advice, including 2 who also received treatment with short courses of steroids. Smell training advice is basically, I've seen people doing this online, which is that you order a sommelier kit of smells, and you basically just sniff it in order to be able to correlate the right smell to the right substance. And apparently over enough time, you can actually retrain your last sense of smell in the people that have lost it for so long. And that seems like that's a psychological activity, which is really odd. Yes, so anosmia is therefore a common feature, emerges several days later than the peak of viral shedding, resolves within 28 days in most individuals. And
I was just thinking it'd be really cool if they were passing out smell tests to people instead of PCRs.
I really do. That would be very cool.
I mean, it seems like it's a really easy thing to send out by mail and people can do it. Seems like it correlates pretty tightly with viral shedding.
I think it starts later than viral shedding, but it could be. It looked like it was a couple days later, but it does correlate neatly. And I would love to get one of those sommelier smell test kits, or even the scratch and sniff smell test kit.
I want the scratch and sniff.
I think you can actually buy those online. Those are only like $30. So if you want to have, a rollicking good time with your friends at a party, smell tests for everyone.
I think we should just talk now.
Is it time? I think it's time. Yeah. They
enough.
basically start to go through there's some
I appreciate that the viral shedding correlates with the PCR structure. So this is very poorly worded, but this figure A or figure 2A is kind of interesting to me now. But it does contradict that paper that we looked at just before this one.
in what sense?
That one showed that PCR remains detectable above the 30 cycle threshold for like half the individuals, even when they were no longer sick. Or I guess they didn't culture their virus, so who would know?
Yeah. The paper kind of resolves in, they're like, look, we really should be using lateral flow assays. At the end of the day, you really should not be referring to this PCR thing, because the PCR thing is putting you in a position where you're going to continuously keep people in quarantine, or you're going to be making interventions for people that do not need the intervention, and that is bad. Because we know that the interventions are damaging. We know that ventilators are bad. We know that a lot of the drugs cause kidney damage. They cause organ damage. Not a good idea. So any way that you can reduce
hospitals are overfilled, stuff like that.
Yeah. There's also like, they kind of end here where they're basically like, so the natural infectious dose of SARS-CoV-2 is unknown. But the virus is understood to be highly infectious. Earlier in the pandemic, the WHO was suggesting that this like minimum infectious dose should be 100 times the tissue culture dose. And they're like, well, we found it that if you actually decreased it by 10 times, that it's enough to meet the target infection rate, which is 50%. But they're like, if you look at all of the other animal studies, you won't be able to clearly correlate between what they're seeing and what we're seeing, because they're using 1,000 to 10,000 times more of the virus in this very aggressive infection model, and that's creating weird dynamics. And, you see the same thing, like you can't compare between influenza papers, because when you do the influenza paper, everybody's already seen influenza, and things are very like tricky and hard to make parallels between. But yeah, I think that kind of covers everything that I have to say about this. We didn't go over this last figure, but this is basically just testing lateral flow assays. And at the very least, the spreads here appear to be a little bit more different than the spreads in the other papers, in the other sections. And down here, they're basically just showing that, you don't have as many false positives as you would with the PCR. Yeah.